Okay, hello everyone. I'm Stuti. For those who have not been acquainted with me yet, I'm a PhD student in Amitesh's group. And as Jonathan was telling, he's not well, so he's not around today. But I'll be talking about a part of what I work on, which is bacterial pathogenesis. And I will try to impress upon you why we look at metabolic plasticity in a bacterial pathogenic context and why we chose pathogenesis as our system to study metabolic plasticity. Okay, so before going into anything else, let me just introduce a little bit about what I do. So I study host bacteria interactions, and these, when we hear words like host bacteria or host microbe interactions, we automatically think about disease states. So we automatically think of host pathogen interactions. But there's another very important context in which you can think about this, which is the human microbiota, which which is what I'm showing on this slide, that roughly the current most recent estimates suggest that there are about as many human cells in our body as many there are bacterial cells. They are very different by weight, of course, because bacterial cells are much smaller than human cells. But looking at the numbers, we are about equal parts human cells and bacterial cells, which makes it very interesting to ask how exactly did the bacterial cells co-evolve with humans and adapt to the human body to be able to survive and thrive within the human niche. So I look at both parts of it, the good bacteria or the commensals and the bad bacteria and the pathogens, and how they interact with human cells and how they live within human bodies. But today I'll be talking about one part of it, which is the pathogenic part. And as I was saying, why metabolic plasticity becomes important in that. So before going further into the talk, let's just get on the same page about what I mean by metabolic plasticity. And this is something that has been coming up over the past 2 weeks, and I expect that most of you have an idea about what we mean when we say metabolic plasticity. But just so that we are on the same page, so let's say I have to do the cell has to achieve a process X, which can be, I don't know, catabolism of glucose, for example, which Sunil just talked about. And you can have two pathways, 1 and 2, which will both lead to the same outcome, which is it will do the process X, right? So these two pathways might be coming from the same place, or they might be coming from different metabolic pathways altogether, but they are ultimately leading to the same cellular process, right? So that's how we think about plasticity. Anything that is plastic means that it can adapt and mold to external factors, and that's how metabolism also seems to work. It can adapt to external stimuli, and then because you have two different pathways that can achieve the same outcome, there's a decision made intracellularly about which pathway to choose. Because even though they're achieving the same outcome, there are advantages and disadvantages associated with each of the pathway. And an example of this was discussed in the last talk as well, where we looked at glycolysis or gluconeogenesis. Those are achieving different things entirely. But if you remember from Amitesh's lecture, and he had talked about the EMP and the ED pathway. So these are both pathways doing glycolysis. So ultimately, they are both leading to the breakdown of glucose, but they have different advantages associated with them. So while the EMP pathway is more efficient in producing ATP, the ED pathway is more efficient in conserving your or utilizing your proteome, or it uses less number of proteins to produce that ATP. So that's what I mean by metabolic plasticity in general. So now that we are clear about metabolic plasticity, I hope, let's talk about pathogenesis. Okay, so pathogenesis literally means how a disease begins and develops. And let's state a very general principle of successful pathogenesis. So to be a successful pathogen, a bacteria must overcome host responses. And we all can agree with that, right? That's a pretty simple concept. So what do I mean by host responses here? So I'll divide host responses into two parts, and I'll call them a passive response and an active response. Okay, so it'll get clear why I'm doing this just in a bit. But by passive response, I mean just the state that the host is in already, that is already determining what is the condition in which that bacteria needs to survive. And then the active response is the immune defense that the host will have once the bacterial pathogen has already invaded that host. So we'll go about it one by one. So let's look at some of the passive host responses. So this is just an example of a bacteria that is invading the host through food, right? So bacterial pathogens that enter the host body through food, so they pass through the alimentary canal, and the alimentary canal basically consists of your buccal cavity, your esophagus, stomach, intestines, and some external glands like the liver and pancreas. And what you can appreciate here is that depending on where the bacteria is situated, it will be facing different types of uh, environments, right? So if we're looking at the mouth, then there's a heat shock because the temperature within our bodies is very different from the temperature in our ambient environments. There's antimicrobial peptides. Versus if you look at something like hepatic tissue, you have reactive oxygen species, you have nutrient limitation, which becomes very interesting. So the one thing to appreciate here is that there's extremely tissue-specific conditions that a bacterial pathogen needs to overcome in order to colonize that tissue. So there's also two types of pathogens. There's pathogens which are systemic, so they cause infection throughout the body, or there are pathogens which are more specific to a particular tissue, but even the specific pathogens will more often than not invade at least two or three types of tissues, right? So that already tells us that there is something in the bacterial pathogen which will allow them to survive under these various conditions. That's clear, right? Okay. So how to overcome this? How to invade in conditions such as this? Okay. It's a slightly complex diagram here, but I'll explain it. So here, the pathways that you see in all of these different things, these are a representation of the energy metabolism pathways inside bacteria. It's a general representation, which means that it's not restricted to any one type of bacteria. Across bacterial species, we see all of these variations of these energy metabolism pathways. And what pathways are exactly represented here? So you have the glycolysis, the ED pathway, EMP, and something called the pentose phosphate pathway. In this area, you have your pyruvate dehydrogenase steps, your TCA cycle, and the electron transport chain. So already you can see that there's a lot of branching present in each of these pathways. In glycolysis, in the TCA cycle, and in the electron transport chain, lots of branches are present, which is the entire idea of something like this, that you have plasticity within each pathway. Multiple different processes are leading to the same similar outcomes. So there's this plasticity. Now, if we look into literature and try to understand which pathways become important for colonization in a particular niche, a particular tissue basically, we see something like this. So the red pathways are the ones which have been shown to be important for a pathogen that is invading that tissue. So for example, for a pathogen that invades the lungs, we see that this particular branch of the TCA cycle becomes important. However, if you look at pathogens invading the colon, then it is the glycolytic pathways particularly that become important. Why is that? That information comes from, once again, this, that what are the components, what are the limitations that the pathogen is facing when it is trying to invade that tissue, and what are the substrates that are present. And depending on that, which metabolism should be used is decided. Right? Okay. So that's about the passive part of it, but that's not it. It's not that a pathogen invades a human body and there's no response actively against the pathogen. We have an immune system as well. So the other thing that the pathogen needs to do is survive the immune defense of the host. Okay, so I'll divide this once again into two types of things. So the active thing is the immunity. So bacterial pathogens can either resist. How far should I not go? Okay, whatever. They can either resist this immune response or they can exploit this immune response. Many a times they do both of the things, and we'll once again see examples of each of those processes. Okay, so this is an example of how you resist an immune response. Okay, so this is a macrophage. So a macrophage is an innate immune cell. What a macrophage does, macrophage does a number of things once it encounters a bacterial pathogen. So there's a cascade of events that happen. But one of the major responses that a macrophage has is called a nitric oxide response. So there's activation of a nitric oxide synthase, and that leads to production of these nitric oxide radicals. Okay, yes. Okay, so a macrophage is so it's a innate immune cell, okay, and it has receptors that can respond to bacterial or pathogens in general, and even respond to injuries. And then this innate immune cell gets activated. It can engulf the bacterial pathogen, and then a cascade of immune responses. It can also lead to the activation of adaptive immunity and so on. Okay. So the nitric oxide synthase produces these nitric oxide radicals. Now, what is the need of producing nitric oxide radicals? So we have been talking a little bit about reactive oxygen species, and I'll come to that also just in a minute. But there's also something called reactive nitrogen species, which do something very similar to what reactive oxygen species do. So just like oxygen radicals, nitric oxide radicals are also toxic to invading bacterial cells because these lead to damage of intercellular molecules like DNAs, proteins. Ultimately, they lead to apoptosis or cell death. So they do this by interacting with active sites of enzymes, which will contain metal centers, like the iron sulfur cluster, for example, and they will lead to the oxidation of those metal clusters, and that's how they interrupt these enzymatic activities. One of the proteins, yes. Is there a mic around? Yeah, at the back. Okay.
Nitric oxide or nitrous oxide radicals, don't they damage the other surrounding cells as well? So why is this targeted only to the pathogen?
They do. So that's why inflammation a lot of times causes tissue damage as well. But you have also, under, let's say, ideal conditions, the bacterial pathogen has to enter inside of the macrophage, and then the macrophage itself gets apoptosized. And the inflammation response should not exceed to such a level that it causes tissue damage. Still, it does, and we see that in COVID, for example, we see that the inflammation is what causes the tissue damage a lot of times. So these things, because it's a general phenomena that they will cause damage to all of these molecules that contain any metal centers, that's why they can cause damage to the tissue in general. Anything else? Okay. So one of the important proteins in bacteria or in organisms in general, which has a metal sulfur cluster, is the cytochrome oxidase. So once again, if you remember from Amitesh's talk, he had briefly talked about these uh, cytochrome oxidases. So you have your membrane, you have your this big thing, which is a NADH dehydrogenase. It gets electrons and it passes electrons to a quinone molecule, and that ultimately passes it to this enzyme complex, which will donate the electron to oxygen, and finally lead to the reduction of this thing. So this is the cytochrome oxidase, and the place where the electron is accepted in this enzyme is an iron sulfur cluster. So these cytochrome oxidases themselves can get affected by reactive nitrogen species. So what pathogens have is two different types of cytochrome oxidases. One is the BO type and one is the BD type. And what people found was that the BD type of cytochrome oxidase is actually resistant to nitric oxide stress. We don't really understand how they are resistant to it. We just know that the IC50 or the inhibitory concentration, 50% inhibitory concentration of nitric oxide for this particular enzyme is higher than that it is for cytochrome BO oxidase. So this enzyme becomes upregulated when Salmonella, which is the bacteria that causes typhoid, is infecting the host tissue. And if you delete this enzyme from Salmonella, then they cannot infect the host tissue to the capability of a wild type strain. So it becomes necessary for their successful pathogenesis. So this is one of the ways by which they can resist a host immune response. You have a question? Okay, you just looked like you had. Okay. So this is how you can resist it, but that's not enough for Salmonella. It just doesn't just want to resist the nitric oxide stress. It would be even better if the cells are able to exploit that nitric oxide stress for their own usage. And that's where one of my most favorite concepts comes in, and this is something called energy axis. This is mostly only studied in Salmonella. So we know about chemotaxis, which is movement towards a chemical stimulus. This is energy taxis, which means movement towards an energetic stimulus. And Salmonella cells do this beautifully. So let me just set up the stage for you for how this will happen. So you have nitric oxide being produced. So your macrophages, okay. So you have this macrophage cell. It encountered a bacterial pathogen and it led to increased nitric oxide production. So that was one type of innate immune response. Another innate immune response that happens in the macrophages is something called a respiratory burst response. Sounds really fancy, it's a really simple concept. So because of the signaling cascade that is initiated by interaction with this bacteria, the respiration of the macrophage goes haywire. And so there's increased respiration. And what we know is that if you do respiration at very high rates, that leads to increased reactive oxygen species as well. So now you have two things that are happening after the bacteria has interacted with the macrophage. You have higher ROS and you have higher nitric oxide. Can anyone think about what will happen here? So you have two types of radicals. You have oxygen radicals and you have nitric oxide, which will also form radicals, but it's also present as nitric oxide as well. So what will happen? Does anyone, any guesses? Pretty simple. Sorry? Apoptosis should happen, but will these two react with each other? They can, right? So these two can directly react with each other. There can be sequential oxidation of the nitric oxide to form nitrate and nitrate. Okay, so these two species form at the site of inflammation, and this is a byproduct of the inflammatory response itself, just in the host. Now, bacteria have terminal oxidases, these things, that can actually utilize both of these instead of oxygen. Okay, so there's a little bit of a different concept here. We have been looking at the respiratory chain in this fashion for most of the course. This is called aerobic respiration because it makes use of this oxygen. When you use anything other than oxygen in this respiratory chain, it's called anaerobic respiration. So bacteria are able to do anaerobic respiration because they can use terminal electron acceptors like nitrite and nitrate. So Salmonella, upon infection, causes inflammation, which almost all bacterial pathogens do. And then it actively swims to the site of inflammation just so that it can utilize these molecules as terminal electron acceptors and perform anaerobic respiration. That's why we call it energy taxes, because the stimulus is that energetic stimulus. And that's how this bacteria not only evades or resists the host immune response, it also actively exploits that immune response. So is this clear, or are there any doubts or confusions? Okay. Okay. So
Stuti,
yes.
when it reaches the region of inflammation, what happens after that?
Okay, so it's actually like it starts way before that. So people think so there's an immune response upon bacterial entry itself, and ideally all of your bacteria, or at least a majority of the bacteria, should get eliminated at that point itself, and that's how you prevent excess inflammation from happening. So it's shown here what salmonella does. There's some variants or some mutants, you can say, which are called small colony variants. These exist across multiple bacteria. So these are the ones that can persist inside of cells without getting detected, because they're not actively dividing. Okay, they just persist inside of cells. And so these later on, once the immune response is calming down, basically, these cells can now become activated, and they swim to that site of inflammation. And once they are at that, so their chemotaxis proteins actually get upregulated, and the stimulus is thought to be the nitrite and nitrate that is formed here. So they move to that site. So it's actually chemotaxis only. So they move to that site, and at that site, their nitric and nitrite reductases get upregulated. So there they can do anaerobic respiration now, and then so this luminal expansion happens, which is a fancy term for more proliferation of the bacterial cells, and then the infection continues like that. They release the nitric oxide radicals and reactive oxygen species outside of the cell in the lumen. Yeah. So this is because there is salmonella. We're talking about the colon. So all of these are epithelial cells in the colon. This is the lumen. So if a colon is a tube, this is like the center of that tube, and these are the cells that line that. So it's released into the center part. That's where most bacteria are found. Yes.
So the enzyme that reduces oxygen there and the enzyme that reduces the nitrogen species, are they the same?
No, they are different.
They're different.
They're
so
different.
the bacteria has both the enzymes.
Yes. Several bacteria have both enzymes. Some bacteria, which are obligate aerobes, will only have the one that reduces oxygen. The bacteria that have both are called facultative anaerobes. So they can be anaerobic if they want, but they prefer an aerobic lifestyle.
We don't have that enzymes.
We don't have those enzymes.
Okay. And what are the byproducts that are formed, and can they be harmful to
not usually. Not usually. There are. I'll come to it in a little bit. There are multiple different terminal electron acceptors as well. Most of the times, they are not harmful to the host. The byproduct. So whatever it gets reduced to, basically, it shouldn't be harmful. Okay. All right. So it seems like that there are respiratory or metabolic processes that exist inside of pathogens that enable them to invade a host, escape the immune response, and exploit that immune response. So it can be a general phenomena within bacteria that you should have this metabolic plasticity that allows you to do that. And that is what actually exists. So this is at multiple levels. I won't go into carbon catabolism a lot because Amitesh has discussed that slightly. But you can divide energy metabolism into, let's say, three things: carbon catabolism, the TCA cycle, and the respiratory chain. Now, for carbon catabolism, you have actually three pathways. So there's something called a pentose phosphate pathway. Won't go into that, just for knowledge sake. You can do EMP, ED, or PPP pathway. Now, the TCA cycle becomes extremely interesting in bacteria. So we canonically think of the TCA cycle as this round thing. And in bacteria, it can be anything from one line to all the way over here in Mycobacterium tuberculosis, something crazy is happening. So there's multiple different branches, and some bacteria might only have enzymes to do one part of the TCA cycle, and that's it. Okay. And this also becomes interesting if you think about a bacteria that is only doing this part of the TCA cycle. And in one of the TA sessions, I had talked about how the TCA cycle provides intermediates for various biosynthetic processes. So bacteria that only has this might become an obligate intracellular parasite because it needs all those intermediates, which might only come from a host. So things like this can also determine the lifestyle of the bacteria. Okay, and now let's come to our lab's favorite part, which is the respiratory chain. So this part here is how we usually think about the respiratory chain. So you have an electron entry point, you have a carrier, an electron exit point, or you can have electron entry carrier, another transport, like another enzyme, another carrier, and then exit. Those are two types of respiratory chains. We don't have to go into the details of that. But if you look at each component of this respiratory chain, so entry, transport, exit, there are multiple different enzymes that can be responsible for electron entry, multiple different quinones for transport, and multiple oxidoreductases for electron exit. Okay, and that's in bacteria. This is all in E. coli, by the way, just one bacteria. We actually don't have a sense of how diverse this is across bacterial species yet, because it's too much. Okay, so this is NADH dehydrogenase, for example, and we have been talking about NADH dehydrogenase for a while. It takes the electron from NADH, passes it on to the quinone. So the substrate here is NADH, but for bacteria, they can use several different dehydrogenases that can have different substrates, like succinate, fumarate, even hydrogen, stuff like that. You can have multiple quinones, ubiquinone, minaquinone, dimethylminaquinone. Amitesh had talked about these yesterday. So these two are your naphthoquinones, and this is the ubiquinone. And then you have the terminal oxidoreductases. And as I already mentioned, you can have the oxygen-dependent ones and the oxygen-independent ones. So the oxygen-dependent ones are these two. Then you have your NAR-GHI, which is the nitrate one. You have one for fumarate, you have for TMAO, DMSO, all sorts of things. So when you look at something like this and you think about this plasticity model, it appears that a lot of these enzymes seem redundant. They're leading to the same function in the cell. Many a times, experimentally, also, if you knock out one of them, the other one gets upregulated, and it seems like it's taking over the function of the one that was knocked out. So, are they truly redundant inside of the bacterial cell? Turns out, no. So, I'll take an example of Mycobacterium tuberculosis to explain how they are not redundant. So, MTB is the bacteria that causes TB, and it forms these very famous structures called granulomas. So granulomas are nothing but, so once, let's go back to the macrophage. So once an MTB cell infects a macrophage, the macrophage apoptosis actually does not happen. So the MTB bacteria suppresses macrophage apoptosis, and instead it makes this cluster where you have aggregation of bacterial cells in between, and you have several active and non-active macrophages surrounding this area. Okay, there's several cytokines and fibrous cells associated with this, but overall this structure is called a granuloma. These granulomas are formed in the alveoli of infected lungs, and this is how the TB progression actually happens. So MTB is a notorious pathogen. It actually has 3 types of NADH dehydrogenases. So now I'm not even saying that they're using different substrates. They're using the same substrate. They're all NADH dehydrogenases. In fact, Amitesh talked about how you have a big one and then you have a small one. So, okay, there's a difference between those two, but they have 2 types of small NADH dehydrogenases. They definitely have to be redundant, right? They're doing the same thing. They're occupying same amount of membrane space. We actually don't really know what they're doing different, but turns out you cannot create knockouts of this NADH dehydrogenase called NDHA. So knockouts of this enzyme are lethal, which means that the bacteria cannot survive if they don't have this enzyme. We don't understand why. However, you can create knockouts of the other two. So if you create a knockout of the second small dehydrogenase, the NDH1, we see that there's a decrease in the NAD to NADH ratio, which is understood because this enzyme will oxidize NADH to NAD plus. So this reaction is not happening. So you see that there's a decrease. There's a decrease in this ratio. Now, because of this, it turns out that Mycobacterium, which are mutants of this particular enzyme, are resistant to the first line drugs which are used against TB infections. How would that happen? Because both of the drugs mentioned here, ethionamide and isoniazid, they act by forming an adduct with NAD+. Now there's no NAD plus free inside of the cells, and so they cannot form the active form of that drug, and so the bacteria becomes resistant towards it. So even though these enzymes sound like good targets for antimicrobials, we actually cannot use them just like that. Okay, so if you delete this one, this happens. If you delete NUO, which is the big one, this one, then you see that there's increased macrophage apoptosis. Initially, that sounds like something bad, but that's actually something good because, as I was saying, that macrophages, once they are infected with the bacteria, they apoptosize, and that's how they end up killing the bacterial pathogen itself. But if you knock out this enzyme, then the macrophage can perform apoptosis. So the mycobacterium has lost its ability to suppress the apoptosis that's happening in the macrophage. Right? Once again, we don't understand why that would happen, why this enzyme particularly is linked with suppressing macrophage apoptosis, but this leads to increased survival of the host. And interestingly, if you delete both of them together, then you see complete loss of virulence in vivo. So people actually started here, and they saw that if you deleted both of them, then loss of virulence in vivo basically means that if you have a mycobacterium that is a deletion mutant of both of these enzymes, and you use that to infect a mice, then the mice survives and does not develop the symptoms of TB. Okay, so they are both necessary for virulence in mycobacterium. However, you cannot do that because the moment you delete this one, you see that they become resistant towards these drugs. Mycobacterium are notorious for being latent inside of host tissues, so they can persist for a long time, and once you take away the drugs, they'll start doing an infection again. All of those complexities come. But I'm just showing this to show you that even though these three enzymes, and especially these two, seem redundant in principle, they are not, because they supply to various different virulence pathways inside of the bacterial pathogen. Is there any question in that part? Okay, cool. So from all of that, what all have we established? So the pathogenic bacteria will face adverse environmental conditions in the human host. The host will actively defend itself also against the bacterial pathogen. It's not just a passive process. But successful pathogens can both survive in the host environment and exploit the host metabolism for their own virulence. And this metabolic plasticity, even though it's central to pathogenesis, but that metabolic plasticity does not mean that these pathways are redundant. Okay, so this is what we have established till now. And now I will expand this concept into a much more broad example, which is, once again, one of my favorite examples. It is this, I guess. I need my notebook for this. Okay. So I'll give you an example of how important exactly respiration is for bacteria, because we just saw that, okay, Salmonella does energy taxes to the site where it can use something to do respiration. But why is doing respiration that important for a bacterial cell? Right? Okay, so we'll come to that. So I'm going to take example of a bacteria called, it shouldn't be written like this. Citrobacter rodentium. Once again, the name is not that important, but what you can just understand is that, so E. coli in humans, a certain type of E. coli in humans causes traveler's diarrhea. And this is the proxy that we use to study that in mice, because E. coli doesn't cause that infection in mice. So we use this organism. Okay, so Citrobacter rodentium causes infection in the gut, once again in the colon. So how many of you are familiar with villi and microvilli. Everyone kind of knows about it, I guess. Okay. So you have these structures in the epithelial lining of the gut, which are the villi, which are important for absorption of nutrients from the lumen, right? So the interesting thing about villi is that the cells which are present here and the cells which are present here are actually metabolically, they are in general really different, but they are also metabolically really different. The cells here are actually stem cells that will proliferate and differentiate to ultimately form the cells that are present at this site. Okay, so they are different, which means their metabolisms are also different. So the cells at the top perform butyrate fermentation. So where did butyrate come from? So in the colon, there's also the commensal gut microbiota, which I was talking about when I started the presentation. There are some bacteria that are just associated with our gut. So those bacteria in the gut produce butyrate, which is used by these cells. It's converted to CO2, but this process uses up oxygen. Okay. The reason why they do that is another separate thing. We can discuss that if anyone is interested. But the cells at the top are performing butyrate fermentation and using up oxygen. The cells here don't do that. Okay. So the environment in the gut is in general hypoxic, which means there's less oxygen concentration. However, because of this reason, the environment here is even more hypoxic than the environment here. There's slightly more oxygen present in the crypts of the villi. Okay. So this is just the background that you need to know to understand what Citrobacter rodentium does.
Stuti.
Yes.
Yeah, so is there a continuous change in these metabolic pathways as you go from crypt towards the
It's
tip?
not
Or
really
is
continuous. Most of the cells here are differentiated epithelial cells. It's just the stem cells here, the Ki67 positive cells, which don't do this metabolism. It's just in the crypts that you have stem cells and they don't do this.
so once they move up and then they differentiate, there's a sudden switch in the pathway?
As they differentiate, they move up.
Okay. Yeah, and then there is a sudden switch in the pathways that they're
The
using.
switching is because they have become differentiated now. That way. Okay. So that's actually a very good segue into what this guy does. So now Citrobacter rodentium will come here, right? And it will actually colonize the cells at the top. Then it has something called a secretory system, which we don't need to get into. But ultimately, it produces certain effector proteins that interact with these cells here, or let's make them over here, right? And it leads to their uncontrolled proliferation. So I'm only saying proliferation and not differentiation now. So they'll just start dividing, and they'll come to the top, right? So all the cells here are essentially those stem cells that were present in the crypt. And because these cells here now are not doing butyrate fermentation, the oxygen concentration here will increase again. Okay. So Citrobacter rodentium does all of this just so that the oxygen concentration increases here. And can we think about why that might be? What's the use of doing that? Pretty simple. Yours over. Yeah, yeah. So they want to respire aerobically. So, okay, so the salmonella was trying to do energy taxes to anaerobically respire, but this guy changes the entire structure of architecture of the villi to do aerobic respiration. And we have learned in Pavlov's talk that oxygen is the most efficient terminal electron acceptor, right? So doing aerobic respiration is even better than doing anaerobic respiration. Any type of respiration suffices, but aerobic one is always better. So it does this, and now with this increased oxygen concentration, it can now divide and proliferate. So what was the importance of respiration here? So let's go back to this part, that you have commensal gut microbiota. Okay, so any pathogen that wants to invade the gut actually faces competition from the bacteria that are already present in the gut. Because the bacteria present in the gut also release certain toxins, they are competing for the same nutrient resources, and so there's competition that the invading pathogen will face when it's trying to invade the gut. And so in this situation, by doing another important thing, sorry, I forgot, most bacteria present in the gut, a majority of them, actually do fermentation. And the reason for that is exactly that the conditions in the gut are hypoxic. So the bacteria doing fermentation will not divide as rapidly as a bacteria that will do aerobic respiration. So the moment Citrobacter rodentium or E. coli in our situation is able to do aerobic respiration because of increased oxygen, it can now outcompete the resident microbiota and then cause an infection in the colon. Was this clear? It's a little complex. Okay, cool. This is exactly what I've shown here. So you have your complex, your microbiota that produces butyrate. It gets converted to CO2 by using oxygen. The cells in the crypt don't do so. And then Citrobacter leads to certain changes in the cells in the crypt here, leads to their uncontrolled proliferation. That's called crypt hyperplasia. That's just the term for it. And ultimately, these cells are able to survive here. Okay. Okay, so why are we studying all of this? Why did we get interested in this? Is a translational aspect of this. So you talk about pathogenesis, and you cannot ignore the antimicrobial resistance part of it. So I don't think I need to convince all of you about antimicrobial resistance and why that's a problem. But this slide is just for doing that. So this is from 2022 in Lancet, and these are all the deaths globally directly associated with or just attributed to antimicrobial resistance. And you see that the number is close to 900,000. The most interesting thing here is that the biggest cause is E. coli, which is the bacteria that we have been talking about and we all use for research all the time. But the major concern in AMR or antimicrobial resistance right now is E. coli. Anyway, so this is the scenario. And there are a lot of groups across the world working on how to tackle antimicrobial resistance. And one of the ways by which people are doing that is by generating new antimicrobials, so novel antimicrobials. The problem is that conventionally, most antimicrobials have targeted essential pathways within the cell, such as cell wall biosynthesis, translation, transcription, DNA replication, and so on. And one area that has been largely ignored up until a decade or two ago has been the cellular metabolism and respiration. Very recently, people have started looking into these as targets for antimicrobial drugs. But it makes sense that you would use these as targets for antimicrobials because they are essential for the cells, for bacterial cells, and a lot of them are actually not present in mammalian cells. So their counterparts are not present. And so they make good antimicrobial targets. In fact, one of the first drugs in the last 40 years that got approved for treatment of multi-drug resistant TB infections was called bedaquiline. And bedaquiline is this diaryl quinoline molecule that inhibits the ATP synthase of mycobacteria. It's extremely specific to mycobacteria and it's extremely potent as well. It's used as a last line drug right now, and it only came into the market about 7 to 8 years ago. And we already have instances of resistance against bedaquiline. Okay, so the production of novel antimicrobials is important, but that obviously does not preclude the possibility of having antimicrobial resistance develop against those drugs. Right? And that's where we come in, and we do adaptive laboratory evolution in the lab. So just a little bit of a refresher about what ALE is. So ALE is adaptive laboratory evolution. So you have your bacteria growing in cultures, or, well, let's just stick to cultures right now. So you have your bacteria growing in cultures, and you provide them with a certain selection pressure, which can be an antimicrobial, for example. And then you passage them for several generations. And then you see that after these several generations, if they're able to overcome the stress or the selection pressure that was provided, then what was the change in the evolved strain that led to that shift? Okay, so that's how we do ALE, and this is one of the recent papers from the lab where we have looked at what happens if you inhibit this smaller NADH dehydrogenase in E. coli. And the reason for why we are interested in this one, it's the major NADH dehydrogenase in E. coli under aerobic conditions, and it's not found in mammalian cells. So it's actually a target for novel antimicrobials. There's several antimicrobials in the pipeline which are acting against this one. So by doing an evolution study upon deletion of this particular enzyme, you can try to understand how E. coli or any bacteria that you're studying will evolve to compensate or overcome the loss of this enzyme. So what is the metabolic rewiring associated with that? So once you know that what is the metabolic shift that led to compensation of this, the loss of this enzyme, you can design a rational drug therapy where you don't not only target this enzyme, but you target whatever happens upon metabolic rewiring after evolution as well. I'm not planning to go into this work. We have a lot of time, but huh? Okay. If anyone has questions about it, I'll go into it. But I was not planning to discuss the entire work in length. Yes. Hmm. It does. It does. Exactly. Yes, it does. So that's what I'm trying to do. So in the lab, this evolution, for example, was done in minimal media containing glucose. And that's a bacteria that will develop resistance against, let's say, a drug that inhibited this enzyme. It will not be developing resistance in minimal media containing glucose. It will happen inside of a human cell, for example. So right now I'm designing an ale experiment where we do evolutions inside cell lines. People in the field also do evolutions inside mice. And so those things are coming up very recently. And people are trying to mimic the natural conditions more or in a better way in that respect. But you're correct, it will depend on the. So that's what the rational, so you can create combination therapies. So it won't mitigate antimicrobial resistance per se. It can delay it, or it can, it in some situations actually. So okay, there's multiple concepts here. So if you have two drugs, if you supply two drugs together, it can also prevent something called collateral resistance. So collateral resistance is when you give one drug, one antimicrobial drug, and because of whatever that drug did, the bacteria becomes resistant to another unrelated drug. Okay. And if we people in the field have seen that if you do combination therapy where you provide multiple antimicrobials together, you prevent this collateral resistance development as well. So in that respect also it becomes useful. So that's the type of work we're doing in the lab. And I'll just end with this little thing. So yesterday there was a lot of discourse about how in biology we don't have set principles or biophysical principles that will determine the cellular processes or metabolic processes occurring inside of cells. And I found that really interesting because that's a very fascinating idea that we can have a set defined rules for metabolic processes and we can understand metabolic processes using those rules. And the example of Maxwell equations was being used for that. And that obviously made me think of the germ theory of disease. So this is something called the Koch postulates. And they're not mathematical postulates, they're actually experimentally derived. Oh, postulates. Okay. Yeah, please don't mind my pronunciation. I'm just butchering his name. But anyway, so yeah, they are actually experimentally defined. But these are postulates, and they are very simple if you look at them. Now you're obviously an organism will lead to a disease, and if you take away the organism, the disease will go away. You introduce it back, the disease will come back. They seem simple, they're elegant, but they are the reason that the field of disease biology actually exists at all, right? So just a fun little thing that connected me to whatever discourse was going on yesterday. And similar postulates exist in the field of evolution, for example, natural selection, and for the genetic code and so on. Just a fun little thing. And that's it. I don't have anything else to say. If there are any questions, because we have a lot of time, I'll take those. But otherwise, we're done. It's actually the reason why they do this. They link it to the ATP that has to be produced for functioning of the sodium potassium pumps. And the reason they do this is so that they can perform efficient reuptake of water from their environment, because water reabsorption happens in the colon. So, yes, right, correct. Yeah. So, okay, I think we're done.